You've seen this already, but the number one question that we get is, how many SNPs will I get? And I used to tell people, well, it depends. Now I'm so tired of the question that I say 42. And then I ask, well, that's not the question you want to ask. What question do you really want to ask? This is the one you really want to ask. How many SNPs do I need? And the answer to that, again, 42. I don't know. What are you trying to do with those SNPs? You have to ask which question you're trying to answer with those SNPs in order to be able to answer this question for yourself, right? All right. So the best advice that I can give anyone who wants to do a project and wants to use GBS as a resource for that project is to stop, and before you submit any samples, ask yourself a couple of things. First of all, it's very, very, very important. Does my experimental design make sense? If it doesn't, you do not have a very high probability of having a successful experiment just because you're using a new technology, okay? The second question you have to ask yourself is, do I have a plan for analysis? And sometimes the plan is, well, I don't have the time or the resources to do this myself. I'd really like to hire somebody to do this analysis for me. If you're going to do that, I suggest that you do that in conjunction with this first step, because oftentimes that person who's going to be helping you analyze your data can give you very, very good feedback on whether or not your experimental design makes sense, okay? And the last thing anybody wants is a data set to fall into their lap from a poorly designed experiment and then have to do something with it. It's not fun for anybody. And as you're going through this process, at every single step in the process, I want you to ask yourself, do my results make biological sense, right? We're scientists, and just because we're doing something new, we shouldn't all of a sudden ignore everything we know about the biology, especially of the organisms we're working in over and over again, okay? So keep on asking yourself, if it doesn't make sense to you, you gotta dig deeper. You gotta take a look at what's going on. So GBS can be complicated for a number of reasons. Some of those are technical reasons, like sequencing error, SNP calling, which we'll talk about in a minute, missing data, large data sets. But really, the biggest reason why GBS can be complicated is biological reality. Biology is messy, and that's only exacerbating the problem of why GBS can be complicated. So I often get the question, is GBS more complicated for non-model organisms? And the short answer is yes, it is. But the reason that it is more complicated is because it was not designed for your non-model organism. So you're going to have to make some adjustments. And a lot of people don't have a reference genome. And there are ways of dealing with that. I'll talk about a non-reference pipeline. There's also people who have absolutely no idea about what's going on in the species as far as population structure or diversity. And actually, GBS can be one way to start discovering what that population structure or diversity is like. Also, a lot of people have access to a reference genome that's not fully assembled or annotated. It's in pre-assembly state or where the physical ordering might be inaccurate. These are problems that we can overcome in various ways as well. And also, people don't always have nice maize inbred lines. A lot of time people are working in outcrossing species and with analyses where this can be very challenging. Okay. So first I want to talk, I don't usually give this talk, so you'll have to bear with me as I describe the unique pipeline to you. Usually Fei Lu, who designed the pipeline, talks about it, but I will give you an overview of what it is. And then I'll go into how SNPs are actually called, and this is in reference to the reference-based pipeline, but it's also applicable for calling SNPs from output from the unique pipeline. We'll talk about that. And then I will give you some examples and usage cases. Okay. All right, so as I mentioned, it was Fei Lu who designed this non-reference pipeline, and he designed it specifically because he was doing a study in switchgrass, and there was no reference genome available. And I'm going to give you the citation at the end of these slides as well. Okay. So typically, this is presented after we talk about the reference pipeline, so you're going to hear a little bit of redundancy as I'm explaining things. But basically, the non-reference pipeline is a very simple modification. Well, I'm sorry, it's not simple. It is a one-step modification from the reference pipeline. And instead of doing alignment of sequence tags to a reference genome, we're going to do a special tag-to-tag alignment that I'm going to describe in a minute. But aside from that, all of the steps of the pipeline are the same. So as we're going through our exercises, just realize that everything that you're doing is going to be the same aside from the physical alignment portion. And that's where the unique pipeline comes in. Okay, so as we talked about, the genome is digested and then it's sequenced using this reduced representation approach, and reads are trimmed to 64 base pairs, and that's what we call a sequence tag. Okay? So we create a catalog of all of these identical 64 base pair tags, and the identical reads then are defined as a single tag. So we might have one copy, we might have 500 copies, but if it's all identical, we call that one tag. Okay? So it's not the same as the reads that are coming off of the Illumina machine. It's just a representation of the unique sequences in those reads. All right. So then what's done is we do a pairwise alignment of those tags. So you align the tags to each other rather than aligning it to a reference genome. But the pairwise alignment is done in a fairly restrictive manner, so it only allows one base pair mismatch per 64 bases. Okay? So you're aligning tags with just a single mismatch, which means that if you have something which is over, that would be 1.5% divergence then, you're not going to align those two tags together. That's not going to be a locus. Okay? And from that, you start to build tag networks. So you have a network of things that are one base pair different from each other. Of course, a lot of that's going to be due to sequencing error. So you're going to have noise there that you want to get rid of to discover your true signal. So in this case, there are 3 different tags, and the red and the blue tag differ by one base pair right here. Okay? And then the blue and the gray tag differ by one base pair as well. But in order to determine which are real tags and which are sequencing errors, the information that's used is the total count, the number of times we see this tag in the data. Okay? So in this case, we see the red and the blue tags in fairly equal high numbers. So we determine that these are true tags, whereas the one that's in gray is only seen 8 times. And this, based on the error threshold, which I'll show you in a second, is then determined to be an error. So when you're creating your tag pairs for SNPs, then the only ones that are going to be used for that SNP are the red and the blue. And you can get all different kinds of networks of things that are different by one base pair. This is a very simple case where you've got 2 tags and they differ by one base pair, but you can get all kinds of things like this, right? And what we want to do is we want to apply this network filter and then keep only networks that have common reciprocal tags and potentially also errors coming off of those tags. So you can see as we move from this sort of set of networks here, you can see there's only common reciprocal tags, and then occasionally you get the sequencing errors. Those sequencing errors will not be used in SNP calling. And those are determined, oh, not the slide I thought it was, okay. So this is actual real data of what a whole bunch of tag networks looks like, and this is in the switchgrass data. So things like this are going to be plastid and highly repetitive tags, and you're moving down to moderately repetitive things, paralogs and SNPs. And so this filter can do a good job at removing paralogs, but it doesn't remove them completely, and I can show you some of that data later. But it's, yes?
When you do the filtering, sorry, on the last slide, when you do the filtering for the sequencing errors,
yes.
are you using these tags from all the different genotypes, or is this just?
Yes, so this is at the population level. Okay. All right, and so there is a user-definable parameter. It's called the error tolerance. It's typically set at about 3% because this was determined empirically to be what the error rate was at that time. And what happens is that you start with a tag network like this and you use those counts like I showed you. And if the number of reads coming from this guy is less than the proportion, oh my goodness guys, sorry. Let me start over. If the proportion of things that are presumed to be error is less than the error tolerance, then that is going to be filtered out. But if not, it's considered a real tag. Okay? So if you have a network like this, then these blue things would be identified as errors. And then what happens to this network, because we don't want to throw this whole thing out if we just have some errors that are contributing to this complex topology, is this gets cut where we see these errors. And so now you're going to end up with actually two different networks, right? You're going to end up with one here and one here. And of those two network types, how many are the ones we're looking for? Is it one? Anybody? Anybody? Is it two? How many? So if I cut this here and I have one network, that's these two tags, and then another network, that's these three, how many of those am I going to keep? One? Anybody? Two? Anybody? Nobody's going to vote. I'm not doing a very good job of explaining this, so I'll just tell you the answer. Okay, so what happens here is, because remember, we're only looking for reciprocal tag pairs, we're going to filter out those complex networks topologies. When the sequencing error is cut off, where we end up with these two different topologies, this is the only one that meets our criteria of being a reciprocal tag pair. Okay, so that one's going to get completely removed. Yes? Things that differ by only one base pair. So two tags that differ by one single base pair. If you've got three in that network, it's not reciprocal. Okay? And then you end up from, remember this really complicated set of networks? From that, after applying the filter, you end up with something more like this. Okay? So one of the things we like to talk about is ascertainment bias. Okay? It's not true that there's no ascertainment bias in GBS. And the problem with ascertainment bias exists in any system, any genotyping platform, right? Even if we're using the reference pipeline, there's ascertainment bias in GBS, and that's because we can't align really divergent sequence tags. So we're missing a really divergent proportion of the genome, right? So that alignment step causes some bias. But the bias is so low that we like to say it's almost nothing in the usage cases that these guys have presented. But you should be really careful, because sometimes when you're applying filters this stringent, like in the non-reference pipeline, you could be, depending on what your sample structure is in your sampling design, in your experimental design, you could actually be introducing a lot of ascertainment bias when you're running these pipelines. So this is something to be very careful of. Okay. So there were some additional experiments done in maize to validate this pipeline. And you'd have to talk to Faye about the details of this. But generally, what he did is he took an allele frequency distribution from maize GBS data that was unfiltered. He applied that network filter that we're talking about. So we go from having a lot of very low frequency alleles to pushing this to a state where we get lots of SNPs that are about 50% allele frequency. Okay? And so he also used LD to validate this, and also ran the reference pipeline. So then it did alignment of these sequence tags and called SNPs, and found that the agreement rate was about 93.2%. So 93.2% of the SNPs were really polymorphic when you use the reference information. And if you want to read more about the study that this was designed for and real biological results, you can read the paper, which is now published in Plos Genetics. Fei Lu is the first author on this, and it's been out for a while, so I'm sure most of you have seen it. Okay. And I guess they haven't seen this yet, have they, Rob? Okay. All right, so you're going to see this a lot later today and tomorrow, but this is just an overview of how the GBS pipeline is run. And this is, again, to illustrate the point that the unique pipeline, as we implement it now, is a very simple fork from the reference-based pipeline. So almost all of the steps are the same, aside from what would usually be right here would be an alignment to a physical genome step. Instead, we're using that tag-to-tag alignment in the network filter. Okay. All right, so again, so SNPs can definitely be called if you don't have a reference genome available, but be very careful with your sampling strategy, as this was not designed for very high diversity species, especially when you've got pardon?
I'm talking to myself. I work in a species where it's about a SNP every 20 base pairs, and you're disallowing tags that have 1 in 64, so I think I have a problem.
We can talk about problems. We'll have a therapy session later, maybe, about problems. So yeah, so definitely. Just kidding. Okay, so be careful with your sampling strategy. Again, this was optimized for a particular application in mind. That application was not a multi-species, high-diversity population genetics study. Okay, and it's as simple as that. It doesn't mean you can't use it and you can't answer questions with it, and I'll show you an example of that later. But be very careful because, like I said, sampling strategy is really going to matter. Allele frequencies can matter. If you've got low frequency alleles in your population, like you saw, a lot of that's just going to be tossed out like sequencing error anyhow, which is true of both pipelines, but again, it's exacerbated when you don't have a reference genome. Okay, so then also decide whether or not your downstream analyses are going to be qualitatively influenced by biases you might be introducing in the SNP calling procedure. What are you going to do with it? Are you going to do association mapping? Are you going to try and estimate population genetic patterns? Do you want to get accurate counts of sequence diversity? These are all the questions you need to be asking yourself before you run the pipeline, get the SNPs, and try and test that. So just to reiterate the point, remember pairwise alignments happen tag to tag with one base pair mismatch. So if you've got greater than 1.5% diversity as a rule, you probably are going to have issues with this pipeline. This is not the only non-reference pipeline available. It's the one that I know the most about. A lot of people are trying Stacks, which has more user-definable parameters than this particular pipeline. So if you do use it and you find it to be useful, it would be great to know that. So sometimes you're going to be identifying the same locus as two separate loci when you've got certain types of population structure and you've got greater than 1.5% overall divergence. And if you've got really distinct subpopulations, sometimes you're going to completely miss loci that are monomorphic within those subpopulations, but are diverged at more than one base pair per 64 bases between the subpopulations. Okay. And again, allele frequency can matter because undersampled diversity in true rare alleles can look like error. And this is, again, it's a problem for both pipelines, but it's exacerbated when you're using the non-reference pipeline. All right. So now we're going to talk about what a SNP is. Can anybody give me a definition of a SNP? And not the words single nucleotide polymorphism. All right. What we're calling a SNP with these genotyping methods is not a real thing. It's a statistical construct. And we're going to talk about why that matters and why it matters more for GBS. So this is a lookup table that is in the GBS pipeline. It's based on the binomial distribution. This is actually when you're calling SNPs, and when we're doing this as an exercise later, you'll see this spit out in the console output or in the log file if you save your output to a log file, which you should. And it's really simple. It's just saying, okay, I have a known number of reads for this genotype. If I'm sampling, for example, this is sampled once, you don't even see it on the lookup table because there's only one option there. If I've only sampled one allele, I have no other information on that genotype. So that's what we're going to call it. That genotype is then that allele, right? But if I have sampled two things there, I now have a choice. Those two things could be the same. Those two things could be different. How do I know if that's an error, if that's real? We absolutely don't know. So we use statistics, and we say, in this case, if I am using a 1% error rate, and that's a user-definable parameter, if I have two total reads, if one of those is different, I'm going to call that a heterozygote. Okay? And we can march on down the table and say, okay, now I have three reads. If I see one allele that's different from the others, I'm still going to call that a heterozygote. And all the way down up until you get 7 total reads, now if I see one, I don't believe it. Now I need to see 2 of that minor allele in order to call a heterozygote, right? If you change the error rate, say, I feel like I have more confidence here for some reason, we can change that whole lookup table. And now we can march all the way up to 8 total reads and say, okay, up to 8 total reads, if I see one minor allele, I'm going to call that heterozygote. Okay? And it's as simple as that. So a SNP is a statistical construct. And I hope to see in the future, yes?
So given that it's a statistical construct and you're going through this, why don't you just assign probabilities?
You stole my next sentence. I would like to see in the future, instead of calling discrete genotype states, that we're calling probability-based genotypes. There's a measure of confidence that we have, and we should take that on down through the pipeline. So thank you very much for making my point. But this is how it is right now, and unfortunately, a lot of the tools that you're going to use for analysis won't accept anything other than discrete genotype states. Yeah? All right, so to illustrate the point about how this can affect you and your GBS data. Since we're sampling, like Rob showed us, we're just sampling a portion of the genome, a portion of the library. What we're doing is we're trying to get as much information at each locus as we can to call a genotype, but we're restricted by what we've sampled. So this is, we're going to try and call a genotype here. And so this is one single locus, and we've got a total read depth of 7. It turns out that there are only 3 distinct tags here. And there's a variable position in this tag. And this is for the reference pipeline, because you can see that there's more than one base pair mismatch. Okay? So we can see that there's a variable locus here. And since our total read depth is 7, assuming a 1% error rate, we're going to say, all right, the major allele count is 4, minor allele count is 3. I'm going to call that a heterozygote. That is an S. There's another variable position at this locus. But in this case, our total read depth is 7, and our major allele count is 6. Our minor allele count is 1. At an error rate of 1%, we're going to call this an invariant site. We're going to assume that this is a sequencing error. Okay? So let's say we ran this library on another lane of sequencing. Okay? And this time we only sampled 3 of those tags. At this locus that we had previously identified as a heterozygote, we have not sampled the other allele in this case. So now this is going to be called a homozygote. What we had previously identified as a sequencing error now is going to be called a heterozygote, right? Because our total read depth is 3 and we see a minor allele. So the reason this is more complicated for GBS is because of what the read depth distribution looks like. A lot of people want to use standard resequencing pipelines and standard resequencing filters. Those pipelines often assume a normal distribution of read depth. Okay? So you can say, if I want to have, and I apologize to the mathematicians because it's probably going to be wrong, if I want to have 90% of my data at 5x coverage, I'm going to sequence to a mean of 20x. And that means something when you have a normal distribution, right? If I tell you what the mean and the median of this distribution are, it gives you some information, right? But who thinks it's going to be useful? Yeah, it's not quite so useful, right? So if you come to me and you say, Katie, I want to do GBS sequencing at depth of 5x. I'm going to say, okay, that's great. It doesn't really mean anything. And I do, when I do analysis for people, I actually do calculate mean and median because you can, like I said, it's not completely meaningless, but with this kind of distribution, it does not mean the same thing as it does here. Yeah. Sorry, go ahead.
You've got this distribution of depth, and I know that's summed across all sides. You obviously must have enough data to look at a single site across all different samples.
Mm-hmm.
And so is it a normal distribution of depth for that particular site?
It depends on the biology of that locus.
But we'll assume it's just a normal diploid species. There's no parallels or anything else. We've got rid of that.
It's not normal.
It's still not normal. Is it Poisson? I should say Poisson distribution.
Now I'm going to be embarrassed because my mathematical skills are not as good as yours. And I haven't actually explicitly tested which distribution it fits best, but it's still not normal. it's still going to look more like this. So, sorry, I can't.
I'll talk to you later.
Yeah, we can certainly, it's not very hard to look at that sort of thing, but right now you're exceeding my mental capabilities for several reasons. Okay. All right, so this could matter to you, whether or not you're able to call heterozygotes, or it could not matter. And in a case where you want to, for example, do phylogeny reconstruction, and you're using distance measures, if you've sampled randomly one or the other allele, it's probably not going to matter for you. I was in a situation where I absolutely needed to be able to call heterozygotes efficiently, otherwise my downstream analyses were not going to work. That was when I was working in grape, and we found that the homozygote undercalling rate was about 30 to 50%, which is much better than it was in maize, which is something, what did you say, 80% heterozygote undercalling in maize, Jeff?
Yeah.
About 80%. So it's because we don't. It's because we actually sequence at a higher plexing level in grape than was done in maize. So we had a little bit more data, but still 30 to 50% is a pretty high error rate when you want to be really confident in those heterozygote calls. All right. So the way that we can approach this problem, we can use several different things. In the maize project, they were able to use genetics, and you saw that with Peter Bradbury's algorithms. Unfortunately, the grape genome is not very good, so we were not able to use that approach successfully. So what we had to do is focus on the high coverage genotypes, and we did this through the creation of a VCF plugin. So when you run the pipeline tomorrow, you see you're going to get HapMap output, which are SNP calls, but it has no associated depth information. We needed to recapture that depth information, and at the time this wasn't implemented in Tassel. Now it's actually being implemented in Tassel, and depth is going to become part of we'll talk about that tomorrow. Never mind. Okay, so the resources for utilizing depth information are becoming more and more available, including in Tassel. All right, so this is what I did. Each project is different, and each project is going to have a different solution. So don't just do what I did. Figure out for yourself if that's what you need. And in order to implement this plugin, there's a lot of help from Chi Sun, Jeff Glaubitz, John Zhang, and Terry Cass-Stevens. And now, instead of having a genotype, we have a genotype, but we have the information that goes into calling that genotype. So we can make a decision on whether or not we want to keep that as something we're confident in or something that we don't have a lot of confidence in. And so this is what a SNP looks like in a VCF file. It's pretty complicated. It's not an A, T, G, C, S, or R, right? The part before the first colon, so this part right here, this is the actual genotype. In this case, 0, 0 is the homozygous major allele. The portion after that, which is 3, 0, 0, are the allelic read depths. So in this case, we have seen the major allele 3 times. There were 2 minor allele states at this locus, neither of which we sampled. The number after that is a total read depth of 3. After that is a genotype quality score that's calculated based on that allelic read depth information. And then these are some reverse Fred-scaled likelihood scores. And you don't really need to remember that. And please don't ask me about the math. I'm going to show you on the next slide. I'm looking at you. So the plugins are available in Tassel 3 to output in the VCF format if you need to do this. And VCF integration is also available in Tassel 4 now directly through the SNP caller. So you can get this information if you need it. The information is available on how to do that.
You just said earlier that distributions.
Thanks. So it's based on the allelic read depth. So if you've sampled one allele at that locus, total read depth is 1. You're going to have a really low genotype quality because you don't have a lot of confidence if there are multiple alleles at this locus. To be in the VCF file, you by definition have to have at least 2 alleles coming out of this pipeline. So if you have also more alleles at that locus, you have to see each allele more times in order to be more confident in it. Okay, and so Chi Sun was the one who developed this pipeline, and he used the algorithm from the Stacks paper, from the Honla paper, in order to call the genotypes. So it's slightly different than that binomial lookup table that we were talking about. But when you compare genotype call to genotype call using these two pipelines, they're largely the same. There are some slight differences. So if you do this yourself and you notice differences between the two, don't panic. It's probably because there are some slight differences in the SNP calling pipelines. All right, so then after obtaining this depth information and applying several different filters, I tried a whole bunch of different things, but here I'm illustrating that filtering on depth alone doesn't get you as far as filtering on that genotype quality, which takes into account the allelic read depths and the number of alleles there. So if I filtered on depth of greater than or equal to 5, I reduced the amount of data in my data set by around 50%. But I still had a fairly high error rate of calling heterozygotes at 8%. But by using a genotype quality score of 98 or above, I was able to retain more information and get an error rate that I found to be suitable. 3% is a slightly elevated error rate here. It's actually lower than that. The reason that's high is because I use real biological data to validate this, and there's error associated with it.
Excuse me?
Yes?
What was the filter on the x-axis on the right? We can't really see it from here.
Oh, so this is no filter.
Yeah.
This is minimum depth of 3,
Yeah.
minimum depth of 5, and this is genotype quality of 98 or greater. Okay?
We get the slides from.
Yeah, I think
Yes.
so.
Okay.
Genus-based quality?
No, that's a very good point. The Illumina base qualities are not used in any of these pipelines at any point.
Okay. And also, did you use the SNP position in the read?
Pardon?
Because
Oh, yeah, so errors are
more likely at the end, and
yeah.
I think some
No,
processing just remove the one that are 10 bases.
we do not do that. We do trim reads, and we throw out any reads. You'll see this as we go through the pipelines tomorrow. But we do throw out any reads that have an N in them. And some people think that's too stringent, but we find that also errors are autocorrelated. So once you get a single bad call in a read, you're much more likely to have other bad calls. And like you said, they're concentrated near the end. So we trim reads, trim them down to 64 base pairs, and throw out anything with an N.
So after afternoon tea, I'll be taking you all through the reference-based SNP calling pipeline with all of these kinds of bits of information.
So as a follow-up, do you then correct your barcodes or not?
Do we correct the barcodes?
Because
As I was,
the barcodes and that they allow for some mismatches.
that's right. So actually that's built. I built that into the molecular biology, but it hasn't been implemented as in Tassel at this stage. So if you know Java and this is an issue you'd like to scratch, we'd probably be happy to have you working on that.
I meant rather you were just saying if there's an N in there, you just the same.
If there's an N anywhere in the read, but also you have to match exactly the barcode and restriction cut site.
Contrast the same, so it's
Yeah.
Yeah.
And there's also,
the correct barcodes.
yeah, and we're going to go through this. There's also situations where you're going to throw away data, and we're going to go through that in detail later.
Yep.
So, okay. If you'd like to work with VCF files coming out of these pipelines, there are a whole bunch of tools already available for you, and I'm sure Rob would love to help everybody use them. VCF Tools is quite helpful, and you can use that program to output in other formats if you want to do downstream analyses in other programs as well. And I don't think that you guys are going to need it here, but I always say to people who say I can't run that program because I don't have Unix environment, it's available on our computing lab, and that's open to anybody. So, all right, so there are a couple of different ways to get VCF output now. You can do it in either Tassel version 3 or Tassel version 4. And I'm just putting this up here so that when you guys take these slides home, you can get this output yourself if you'd like to. So I often also hear from people, I have a reference genome, but it's a bad reference genome, so we don't want to use it. Well, beauty is in the eye of the beholder, I suppose. So I always ask them, how less than perfect is your reference genome? Because even relatively short contigs are suitable for anchoring 64 base pair tags, right? But there are often a lot of assembly errors. So if you have assembly errors, you can have problems that are not going to be alleviated, and even if you had very big long contigs, you're not going to get a good result. And actually, a lot of the errors are made when you're going from short things to longer things. So in some cases, that can be the more fully assembled contig could potentially be worse than the thing that's in a whole bunch of contigs. So yeah, and this is just a cartoon of when you have biological reality of paralogous regions, and during assembly, you can actually collapse those into a single contig. But SNPs can definitely be called with the reference pipeline, even on genomes that are not fully assembled. But there's a little trick to doing this. So if you have, the way that the pipeline runs, if you have 10,000 different contigs, each one of those is going to be treated like a chromosome, and you're going to get 10,000 HapMap files, which most people don't like. And it takes a really long time to run. It turns out that even creating an empty HapMap file has a lot of computational overhead. So it could take you months to run the pipeline, which is not what we want. So there's a very simple fix to this. And you can concatenate those contigs into pseudo chromosomes. But you just have to be sure to pad in between those sequences with ends something longer than what you're trying to align, so you don't align to the junctions. And don't go over 2 billion base pairs in a single pseudo chromosome because of the Java definition of a long, right? Is that right? I never get that right.
It's a Java memory allocation thing.
So if you go over 2 billion base pairs, you're going to get an error. So if you have genomes bigger than that, just go to multiple chromosomes. All right. Okay, so now I'm going to actually talk about real data, things that I did, rather than just scaring you guys. This might scare you too, I don't know. But the first one I'm going to talk about is a non-model case study, and I've already talked a little bit about it, but this is in Vitus, which is a highly heterozygous outcrossing species, and in the context of a multi-species breeding program. So we had some problems. So I worked on this in the context of a project called Vitus Gin, which is a collaborative multi-institutional project where they developed strategic breeding populations. So we had 37 different breeding populations. And we were genotyping both by GBS and SSR. The reason we were doing both is because there was a really steep learning curve and comfort curve moving away from one technology to the other. So we wanted to support the breeders in both getting the low-hanging fruit with technologies that they already know and developing this technology for them in the future.
So I just want to piggyback on what Katie's saying here. This is the kind of thing that we have to think about is, some of us can be really excited about, and it's clear to us that some of these new technologies really could benefit our breeding programs, right, or our research. But if we fail to recognize that some folks are going to be uncomfortable with this and fail to have a plan on how we can deal with that kind of situation, then we're not really going to be helping all that much. So it's great to learn how to use these technologies and everything, but we have to think about it in the context of use. And Katie's example here today really illustrates that kind of situation, and I think many of us will find ourselves in that kind of situation. So let's kind of be mindful of it as we go along.
And if you want advice from the trenches over beers, I'll share that in a less formal context, but it was challenging, to say the least. So they also, in this project, were trying to be smart about the way that they were phenotyping, because oftentimes we know it's really hard to phenotype. Number one, it's really hard to phenotype. Number two, it's very hard to get consistent phenotyping, especially across different groups. So we had centralized phenotyping of biotic stress resistance, abiotic stress resistance, and fruit quality. So we had wine grapes, table grapes, and rootstock grapes in this breeding program. And we wanted to target a few really important phenotypes. And so this is how we were doing it centrally. There were also, within each breeding program, a lot of phenotypes that we were looking at within each program, but these ones were centrally phenotyped. And we also included 32 different wild and cultivated species to help in map building and genome assembly and just trying to figure out things that are going on in the germplasm. And I'll show a few of those things now. Okay. So the first challenge I had to deal with was trying to get dense maps from GBS data in this outcrossing species. And this is a situation in which I absolutely needed to be able to call heterozygotes efficiently because our reference genome is not very good. And so what you can do, and in this case what I did, is I first focused on using Pseudotest Cross markers. These are things that are heterozygous in one parent, homozygous in the other. This is the reason I needed to be able to call heterozygote genotypes. And another thing you could do is leverage deep sequencing of your parental genotypes in order to phase markers. You can also do things like filtering by Mendelian errors and linkage disequilibrium in these kinds of populations, really smart filters to do. And then I'll tell you about the method. So now I'll tell you about the method that I developed in Vitus. And again, I'm using, so for people who aren't familiar with these 4-way crosses, a Pseudotest Cross marker looks like this, where you're homozygous in one parent, heterozygous in the other. But in reality, in this type of cross, you have a lot of different marker types. This is really complicated. So in this case, I simplified the problem down to something that was easy for me to digest at first, which is these types of markers. And these are actually the majority of the types of markers that we find. So I developed a pipeline that I'm still trying to come up for a fancy name for. It's not unique, it's not fill-in, it's not gap-it. I don't know what it's going to be, but we'll see what it happens to be. So this is actually 3 separate pipelines that we developed. 2 of them require a reference genome. These are synteny-based pipelines. So this would just be using physical order, right? This is assigning things to a linkage group based on the chromosome, but then doing genetic ordering within that chromosome. And then the third is a completely de novo assignment of linkage groups. It doesn't rely on the physical genome at all. And then doing genetic ordering within those linkage groups. And we were challenged with the fact that we didn't always have parental genotypes available, so I needed to develop this method that doesn't rely on parental genotypes, can handle, in this case, up to 50% missing data per SNP locus, which is suitable for GBS data, and also a method that could handle lots and lots of markers. So in this case, I was able to put about 26,000 markers on the map for a single biparental family with 200 progeny and grape. Okay. This does create separate maps for male and female parents.
Did you guys digest the enormity of that project right there that she just said? It sounded simple as she said it. But
so
this is what Katie's done here is really, she solved a whole series of problems to reach this goal, none of which were particularly trivial.
No, it was quite difficult,
okay?
actually.
I want you guys to just take a step back and think about this a minute.
Yeah.
Because it's
so we were really happy with
but wait a second,
Don't be
Mark.
scared.
Wait a second, Mark. So I mean, I said this was going to, probably scare us all a little bit, right? But look at the hope here, right? Because Katie has, was faced with this challenge with these large, funny data sets. She understood the genetics system she was working in, right? And came up with some solutions to these problems. I think this should give us all hope that we can do these things, right? But the only way she could do it is through really, really understanding what the heck was going on.
Yeah, I think I spent 6 to 8 months on quality control of the mapping populations themselves when I started this project, before I even started any of the methods development. And that was really important because it turns out that breeding is messy. And this data, yeah, I know, I didn't know that. I wasn't in agriculture before I started this project. But it turns out that I actually, that time was well invested because I learned a lot of things about what I need to do to make this database suitable for these pipelines. So
and this kind of goes along with what we were discussing a bit this morning about building in building in the ability to test things and to find mistakes. I mean, Katie had to build a whole suite of tools to find other people's mistakes.
yeah, it turns out. It turns out when you try and use linkage to define something in a biparental mapping population that actually has more than 2 parents, it's a problem because you don't get the expected linkage patterns. So that's
There you go.
that was one really important problem that I encountered. Okay, so
Don't forget the reference genome, because to my understanding, grape has 2 reference genomes in the public domain. So why do you say there's no good reference genome?
the ordering, so actually just at this most recent PEG meeting, there was a new version of the 12X Pinot Noir reference that was released, and the ordering is much better, and I'm going to show you a slide about that. Anyway, I'll show you a slide about that in a minute. But the problem wasn't that we didn't have a reference genome. The problem is that the ordering of the markers within the reference genome is not at all biological reality, especially because we have very wide genetic crosses. We're working on a multi-species project here. So the Pinot Noir reference genome was not representative of the ordering of the markers in the genome like it was for maize. Okay, so, and we also knew that a lot of people are going to be dealing with this same problem, so I wanted to develop a pipeline that was robust to that and that doesn't even require a physical reference genome at all. Okay, all right. So I guess my take-home messages are, when you're designing your study, please know what you need for your analyses before and while you're planning your project. And sometimes, I know everybody wants to get things going very, very quickly, but sometimes it takes a little while to get the resources that you need to develop these projects and to develop them well. And I think that Rob's doing a really good job of bringing you guys all together in the context where you should be super successful with these types of projects. And the other point is that GBS is a really powerful reduced representation approach, but it has two very distinct components. It's the molecular methods and then also the bioinformatics pipelines. It could be, and both of these are tailorable. So it could be that maybe the molecular methods as they're implemented in the current protocol work really well for you, but you're going to need to develop some new bioinformatics pipelines, or vice versa. So also the GBS bioinformatics pipelines were developed and optimized for a very specific application in mind, so they can work for you, but be really mindful of what your needs are for your project. So keep calm, and don't be afraid to filter. A lot of people say that they don't want to throw away any of their data. But we need to separate the wheat from the chaff, thank you. Again, I'm not really an ag person, so.
That's all right, Katie, we forgive you.
The good beer from the bad beer, no, I'm just kidding.
There you go.
I used to be a yeast biologist, not an alcoholic. So you can expect to filter maybe up to 90% of SNPs when imputation isn't practical. And that's okay, because what we're left with is a good, robust data set, hopefully, if you've done your filtering correctly. And you can do lots of pedigree-independent filterings if you don't have structured populations to assist you in filtering. You can, like I said, use read depth and genotype quality. You can use percentage of absent calls, so you can filter down to things that don't have a lot of missing data. You can use minor allele frequencies. And for inbred species, inbreeding coefficient filters out most prologous sites, like we were just talking about. It's really powerful. But you're likely to require customized bioinformatics, especially if you don't have a reference genome. So also be ready for post-processing of data into other formats. And if it's something you have to do often, either code it yourself, get somebody to code it, and then share it. And really just have a plan. So I'll leave you with hopefully this very good advice and take any questions.
Got a lot asked during your talk.
Yeah, that was good.
That was great. I'm really, we're seeing things are starting to ramp up in here with audience participation, which is always wonderful.
Everybody always
Katie.
looks a little bit uncomfortable when I get done, though.
Yeah. So I want to just, we're running a little bit long, but I want to summarize a few things from what we've done so far into a kind of a simple, somewhat simple concept. You know, we've talked about tools this morning. We've talked about tuning a little bit, as Katie just mentioned, both with the molecular method and with the bioinformatics, right? And when I start thinking about this system, I think, you know, it's like this box with a bunch of knobs on it. And you can turn this one this way and that one that way and this other one this way. And, you know, once you understand what these things do, you can really focus in on, you know, how to make the whole of it applicable to your question. And most of them are, and this is a key point, most of them are knobs that you're turning, not just switches where it's on and off, right? You've got to test things and think about it. And I think the other couple things that came out so far today are there are lots of different resources you can bring in to your data analysis, right, and your project planning. So Jeff spent a lot of time talking about things with a robust reference genome and many, many, many genetic resources, right? Katie just gave some examples of things with not, you know, no reference genomes, not great reference genomes, utilizing transcriptomes. So there's lots and lots of pieces that we can work with in this genomics era that can kind of interact with this what I thought was a relatively straightforward technology that I developed, is it turning out not to be the case, right? But the cool thing is that we can understand it. We can really apply it to many different questions. And I think the other thing that both of these last two talks show is that you can, by really understanding your science and your questions and the data, you can develop new methods to answer things that people have never done before.